Great, thank you for the introduction. Good morning, everybody. Shoreline is a local biotech here in San Diego that's using iPSC-derived technologies to develop differentiated, effective, multi-edited iPSC-derived allogeneic innate immune cell therapies. That's a mouthful. Let's dig into what that all means. Disclosures. So over the past 2 and a bit years at Shoreline, we've been developing technologies, acquiring technologies, and developing the infrastructure and the team required to use those technologies to generate transformative treatments for oncology. We believe that we need to make the right edits in the right cell for the right indication to be truly successful in this space. When we think about the editing, we've acquired premier genome editing technologies from our collaborators at Editas Medicine, some of the pioneers in this space, and 2 technologies in particular, an enzyme called CRISPR-Cas12a and an insertion technology called SLEEK, which allows us to do highly efficient gene knock-ins and also knockouts. Once we have those edited iPSCs, we can then generate clones, characterize those clones, bank those clones, and differentiate them into a variety of immune cells. At Shoreline, we're interested in differentiating these cells into iPSC-derived natural killer cells, NKs, and also iPSC-derived macrophages. And by generating a balanced portfolio with different cell types, we're allowed to go into, or enabled rather, to go into different indications where we see these cells being most effective. And over the years, we've generated a pipeline or portfolio that includes combination of wholly owned programs and also partnered. To make the technology into a drug, you need a team of people, and we've also developed a fantastic group of scientists and drug developers that can take these technologies, make them into products, and advance those to the clinic in a speedy fashion. You also need the capital to invest in all of this and fund it, and we are well-resourced to execute on our mission over the next couple of years. We have a top-tier investor syndicate with some household names that I'm sure you'll recognize, but we also have a fantastic array of strategic partnerships with pioneers such as Kite, BeiGene, and Editas, pioneers in cell therapy development, protein biologics development, and genome editing, all of the components that you need to make a successful cell therapy company. And these partnerships have synergistic expertise that have enabled us to get a smooth start as we run towards the clinic. We're also led by an experienced management team and who have a proven track record for developing drugs in oncology and delivering those to the clinic. You'll recognize the faces up behind me. You may also recognize the names, but you'll most definitely recognize the companies that these people have worked at over the years. And together they bring in expertise in iPSCs, cell manufacturing, gene editing, and immune oncology drug development. We're also supported by a top-tier scientific advisory board that we meet with fairly regularly. And when any of these people can't make it to a meeting such as today, you're left with me, but that's okay. Because we can take some time this morning to talk about the science that we're developing here at Shoreline and what we're trying to do with it. And really need to start by taking it back to the core of iPSC cell therapy technologies and what's the most important thing. And that is the ability to engineer those iPSCs in an exquisite way to generally have a cell at the end that has all of the features that you want. So we've acquired this technology through a collaboration with Editas Medicine, again, pioneers in this CRISPR editing space. And we're using an enzyme called AsCas12 Ultra. And this enzyme has been engineered and evolved by our colleagues at Editas to have higher fidelity, up to 100 times higher fidelity compared to standard Cas9 or CRISPR editing enzymes, but also have higher efficiency. And this combination of high fidelity and high efficiency makes, we believe, Cas12a Ultra to be one of the best CRISPR editing enzymes that's currently being used in cell therapies. But having a great enzyme isn't enough. You also need means of using it and using it in an efficient manner. And we're adopting a second technology from Editas called SLEEK. Now, this stands for Selection by Essential Gene Knock-in. And what this technology does is creates a cut in the terminal exon of an essential housekeeping gene, for instance, GAPDH, and we then target our gene knock-in constructs into that location and that vector has the sequence to reconstitute the disrupted essential gene. So if you have cutting and no insertion, the cells die in the time it takes for GAPDH or whatever gene we're targeting for the protein to turn over. But if you do have successful insertion and you do have reconstitution of that essential housekeeping gene, then those cells will be enriched. And what this leaves us with is a much more efficient way of creating pools of highly engineered iPSCs that we can then select clones from. And our colleagues at CRISPR have shown that you can do this to knock in several genes into one location. There are many locations around the genome that are amenable to this as well. And we at Shoreline have also shown that we can do this now and are continuing to develop this technology further. And what this fantastic gene editing technology allows you to do is to really think about what is the perfect cell that we want to create for the indication that we're interested in. For instance, solid tumors. And at Shoreline, we've been using SLEEK and Cas12a together to engineer cells to have greater tumor homing, knocking in synthetic or recombinant chemokine receptors that either complement or further enhance the ones naturally expressed by the cells. We're also obviously looking at tumor targeting through either CARs or complementary ADCC or ADCP receptors that enable CAR-mediated or antibody-mediated targeting of tumor antigens. As we're going after solid tumors, and as many others in the field are thinking about, you need to be armoring your cells. You need to be defending against the immune suppressive tumor microenvironment that they are entering into. And we're using this technology to knock in defense receptors or cytokine support, and also to knock out checkpoints and pathways that would normally be counterproductive for tackling a tumor. And finally, as this is an allogenic product, we're interested in looking at immune stealth, not only to enable better delivery of our product without the requirement for loading lymphodepletion, but also to enable multiple dosing, which we believe is one of the key benefits of allogenic cell therapy, the ability to dose the patient multiple times, maintain that high Cmax, and drive cures. So I'd like to spend a wee bit of time this morning now talking about one of the programs that we're developing at Shoreline utilizing this technology, and that's our iPSC-derived macrophage program. Now, macrophage is a really interesting cell when it comes to oncology, because of all the immune cells, they actually get into solid tumors very easily. And if you talk to anyone developing CAR-T or other cell therapies for oncology, what's your number one problem? It's getting the cells into the tumor. And macrophages, we know, can get there because they're already there. They're one of the main immune cell populations that you find in most solid tumor biopsies. However, when they're there, they don't normally do what you would like them to do. They just kind of hang around, and in fact, sometimes they're counterproductive and are actually driving tumor growth. So what we're doing at Shoreline is developing engineered iPSC-derived macrophages that can take advantage of this penetration of the solid tumor, and then once they're react to the tumor cells through CARs or antibody targeting, start to kill the tumor through phagocytosis or other non-phagocytic means, and then produce cytokines and chemokines that can recruit and activate the broader immune response. Because I'm sure, as we all agree, it's not an immune cell, it's an immune system. And to get to drive holistic, complete cures in cancer, you need the complete immune system. So we have a cell here that can not only target cancer itself and start to eat the cancer cells, it can also recruit and activate the other immune cells that are required to finish the job. However, one of the major issues with macrophage cell therapy is that these cells are very difficult to engineer, and they don't proliferate. So achieving a highly engineered product that's going to work in the clinic and getting enough of those cells to dose a patient once, let alone several times, is actually quite challenging. But by using the iPSC approach, we can engineer our cells at the iPSC level and then expand those into billions and billions of iPSC-derived macrophages that can then be cryopreserved and ready to be thawed and dosed when the patient needs it. So to provide an example of some of the ideas we have around this technology, the first slide here we're looking at is SERP-alpha knockout IMAX. Now, SERP-alpha is one of the famous checkpoints of macrophages, right? it recognizes CD47 that's often highly expressed on tumors, and CD47 binding to SIRP-alpha on a macrophage deactivates the macrophage. It's very similar to the PD-L1, PD-1 checkpoint pathway in T cells. So by removing SIRP-alpha, we enable these cells to unleash their full potential against the tumor. And in the middle here, in the top, we have some in vitro data looking at macrophages killing Raji cells, a B-cell lymphoma cell line. These cells express CD20, and we were culturing our macrophages with rituximab in this case, an anti-CD20 antibody. And what you find is that by knocking out SIRP-alpha, you greatly improve the killing efficiency of the macrophages, and we're seeing a 1,000-fold increase in activity by knocking out this one gene on its own. Now I'm going to pause here because it's really important that that video actually worked, because in a couple of slides I'm going to talk about machine learning and AI, and if we couldn't get the video to work on the slide, you might not believe me. But what we're looking at in this video is the IMAX targeting a solid tumor cell line, in this case BT474, breast cancer. These cells express HER2. We're combining our macrophages with trastuzumab, an anti-HER2 antibody, and what we see on the left is the wild-type IMAX can kill solid cancers, especially BT474s. However, by knocking out SIRP-alpha, you greatly increase this killing. And we're also seeing this in vivo, where if we use a model, a Raji model this time of B-cell lymphoma, and pre-complex our macrophages with an antibody before dosing into the mouse, we see that we can not only kill the tumor in the mouse, but that this is enhanced by inhibiting CD47. We're also exploring this with CARs, and we've generated data now with SIRP-alpha knockout CAR IMAX, and we've been pushing these to see what is the full potential of a CAR IMAX, especially when you combine it with a SIRP-alpha knockout. And on the left here, we're looking at some serial killing data, where we're plating our macrophages down, and every couple of days, adding more tumor. And what you can see here with the green and red lines is that every time we add the tumor, within a couple of 24 hours, the tumor's completely gone. And I want to bring your attention to the scale of the axis at the bottom. This isn't 4 days or 4 rounds, it's actually 4 weeks. And these cells can kill in culture for up to a month. And what we're seeing is that the SIRP-alpha knockout here is providing an advantage as well. On the right, we're looking at some serial killing assays where if you have a wild-type CAR IMAC, the serial killing is not very good, even after 250 hours. But if you have the SIRP-alpha knockout, it's greatly enhanced. So to wrap up, I'd like to talk a little bit about the smart manufacturing principles that we're adopting at Shoreline, and have really tried to implement in our thinking and as we go on, our doing from day one. And really, this is the recognition that there is a world of technology out there, and cell therapy is actually quite old-fashioned. A lot of it's done by hand, the technology is not very advanced, and when you compare it to the rest of the drug development world, we're behind. So at Shoreline, we've been trying to implement these technologies earlier, not only in the manufacturing of our products, but also in the initiation of programs. And we're using AI and machine learning algorithms to help guide the way that we edit our cells, and also to help guide how we design what we put into those cells. We're using automation to handle all of our gene editing, our cloning, our screening, and maintenance of iPSC lines. And we're also using automation to take those iPSCs, differentiate those into terminal effector cells, NKs and macrophages, and then run assays on them. Just increasing the throughput and the quality of the data from the start of the process, and reducing the cost of product iteration and design. And then when we have our product, we're now adopting automation in our manufacturing, and to not only scale up, but actually control the whole process end to end, from iPSC in to cryovial out. So thank you for your attention this morning. I'd just like to leave you with some final thoughts. At Shoreline, we're really trying to build a legacy biotech company here that's engineering cells for cell therapies for serious diseases. We're doing this by integrating state-of-the-art gene editing with premier iPSC-derived effector cell engineering and development. And we're adopting automation and smart manufacturing principles from day one to enable this and make it quicker. And ultimately, this will allow us to create the next wave in cell therapies, but not only iNKs, but also IMAX. If you'd like to know more about the company, please do grab me before the end of the meeting. Love to chat about this further, but if not, please visit our website. Thank you very much.